Hello, everybody, and welcome to a new episode of the MDS podcast, the official podcast of the International Parkinson's and Movement Disorder Society. I'm your host today. My name is Eduardo Fernandez from the Centre for Preventive Neurology at Queen Mary University in London. And we are going to discuss today a recently published paper on the use of proenkephalin as a biomarker in people with Huntington's disease. I have the honor to have Dr. Mena Farag, Clinical Research Fellow from the Huntington's Disease Center at UCL Queen Square Institute of Neurology, who is the first author of this paper. The paper is titled Cerebrospinal Fluid Proenkephalin Predicts Striatal Atrophy Decades Before Clinical Motor Diagnosis in Huntington's Disease. Mena, congratulations on your paper and thank you very much for joining us today.
Thank you for the invitation and for having me on the Movement Disorder Society podcast, Eduardo.
So just before we dived in your article, what is proenkephalin and how is that linked to the pathophysiology of Huntington's disease?
So we know that clinically, Huntington's disease presents as a triad of progressive motor impairment, cognitive decline, and neuropsychiatric disturbance. With age at onset, typically in mid-adulthood, though with considerable inter-individual variability. And at the neuropathological level, however, Huntington's disease is defined by early and regional selective neurodegeneration of the striatum. And medium spiny neurons represents the most vulnerable neuronal population, and striatal atrophy is among one of the earliest and most consistent imaging and post-mortem findings in the disease. And within the striatum, neurons of the indirect pathway are particularly susceptible. These neurons are characterized not only by their GABAergic phenotype, but also by expression of enkephalin-derived neuropeptides. And so proenkephalin, or PENK, is the precursor of enkephalins and is highly enriched in the indirect pathway projecting to the globus pallidus of the striatum. And both animal and human transcriptomic data demonstrate that PENK expression is concentrated within the basal ganglia, and that supports its biological specificity to the striatal circuitry. And prior to this work, post-mortem studies in Huntington's have shown reduced enkephalin content across basal ganglia nuclei, which suggests that there could be early dysfunction or loss of enkephalin-expressing neurons. And these findings really provide a strong mechanistic rationale for PENK as a candidate biomarker of striatal vulnerability in HD. Over the last 5 years, there's been cross-sectional studies that have shown that PENK can be reliably quantified in cerebrospinal fluid, and that CSF PENK concentrations are consistently reduced in Huntington's disease. And these reductions have actually been replicated across independent cohorts, which supports its role as a candidate disease stage biomarker. And of note in neurology, and compared to other neurodegenerative diseases, CSF PENK levels were lower in people with Huntington's compared not only with controls, but also with other neurodegenerative conditions, including Parkinson's, Alzheimer's, and ALS. And importantly, these lower concentrations of PENK have been shown to correlate with disease burden and motor changes, which also supports its biological relevance to the pathology of HD.
So it sounds like you have evidence for what sounds like an ideal biomarker. It's specific, it's linked to the biology, it changes over time, it's correlated with the clinical changes. And that's what your study tries to demonstrate, whether this is more a specific biomarker. And in your study, your hypothesis is that it may be more specific than other markers of neurodegeneration that has been used before in people with Huntington's or more generally in neurodegeneration, such as neurofilament light chain. And also you compare that with progression on the MRI and also progression on the biological staging of Huntington's disease. Can you tell us a bit more about the hypothesis and the outcomes of this study?
So despite the signal that's been quite promising in the studies so far, there have been some important gaps. Prior studies were largely cross-sectional. They had relatively small or predominantly clinically manifest cohorts, and they actually didn't address whether CSF PENK changes longitudinally or relates to regional brain atrophy. And in particular, the actual neuroanatomical specificity of CSF PENK as a marker of striatal neurodegeneration had not been tested. So really, the aim of the study was to evaluate CSF proenkephalin as a biologically informed biomarker of early HD, focusing specifically on the far from onset HD young adult study cohort. And so we aim to investigate both cross-sectional and longitudinal associations between baseline CSF concentrations of PENK and regional specific neurodegeneration using structural MRI. And so our main hypothesis was that lower baseline CSF PENK concentrations would be associated with subsequent brain atrophy, predominantly in the striatum. And we also, like you said, sought to compare the regional patterns of PENK-related structural associations with those observed for other biofluid biomarkers that showed disease-related group differences, mainly neurofilament light, which we know reflects more global and generalized neuroaxonal injury. And we talk about staging and the potential for this work to have relevance for stage stratification and clinical trial design. And so this might be a good point to introduce the HD integrated staging system, or HDISS, which is a key framework which underlies the relevance of this work. And traditionally, staging in HD has been grounded in clinical phenotype, so broadly distinguishing between pre-manifest and manifest disease. And while this is clinically useful, this approach does not really capture the underlying biological progression that precedes the symptoms that developed. And so the HDISS was developed to address this limitation by providing more of a unified, evidence-based framework that captures the natural history of HD across the whole continuum. And so there's 4 stages in the HDISS, and the work in this paper basically includes participants from the HD young adult study who were at stage 0, which included participants that were gene expansion carriers with full penetrance but no sort of detectable biological changes. Stage 1 is actually defined by biomarker evidence of pathology, and it's categorized specifically by volumetric MRI measures of caudate and putamen atrophy. And stage 2 captures emergence of subtle motor and clinical signs, cognitive signs, and stage 3 reflects functional decline. And a lot of the clinical trials at the moment enroll participants that are in HDISS stage 2 and 3, and so early manifest disease. And so a lot of this work and a lot of the work for the HD young adult study, the whole premise of it really is to track the earliest biological changes at earlier cohorts that could potentially be used to inform preventative trial design.
That's an excellent summary, and that's very interesting that your study is focusing on this very interesting cohorts at the very earliest stages of the disease process. Can you tell us a bit more about the study cohort and also the methods that you used in the study?
Of course, the HD young adult study is a longitudinal observational cohort of young adults who carry the Huntington's disease gene expansion and on average are about 20 years from predicted clinical motor diagnosis. And they were matched alongside unaffected controls, very closely matched in terms of age, sex, and education level. And so actually the study was conceived and led by Professor Seri Tabrizi, and it started in 2017. And there's been so far 2 time points. We've recently finished the 3rd time point. And the interval between visit 1 and visit 2 was approximately 4.5 years. And so the cohort is really deeply phenotyped. So it included multimodal assessments, including biofluid sampling of both blood and spinal fluids, imaging, so with 3 Tesla MRI, clinical examination, cognitive testing, and neuropsychiatric profiling. And we've had, over the 4.5 years, we had 103 participants return for follow-up, and we also recruited 23 new participants. And in terms of the methods, particularly in this paper, it's predominantly image biofluid-based analysis. So to analyze brain structure, we used whole brain voxel-based morphometry, which is known as VBM. And so this is really, so rather than pre-selecting regions of interest, this approach allows us to assess grey and white matter volume across the whole brain in an unbiased approach. And it involves segmentation of the brain into grey matter, white matter, and spinal fluids. And we used established methods, VBM methods, to quantify within subject change over time in the brain. And on a voxel level, it allows us to basically see maps of local tissue expansion or contraction between the first time point and the second time point to identify where neurodegeneration is occurring and what rates. And then what we did was we tested the first time point level baseline fluid levels of proenkephalin in CSF and also in plasma and CSF NFL. And we used those to predict where the spatial pattern and magnitude of atrophy were found, and we adjusted this for age, sex, the time interval between scans, and also the disease burden. So the key point is really that we're not just asking where do these groups differ at a single time point, but we're asking where in the brain tissue loss is occurring over time and whether baseline biofluid biomarkers can predict the spatial pattern and magnitude of that loss.
And did these biomarkers show the pattern, the anatomical pattern of atrophy on MRI?
So the main finding of the study was that baseline CSF PENK strongly predicted brain atrophy with a predominantly striatal spatial pattern. Specifically, lower baseline CSF PENK concentrations were associated with greater longitudinal grey matter loss in the caudate and putamen on both sides, with corresponding changes in white matter in the adjacent peristriatal white matter. And these associations remained significant even after adjusting for the disease burden, which indicates that PENK provides information beyond age and CAG repeat length alone, which we know are known drivers of progression in Huntington's. Whereas NFL showed quite a different spatial profile. Higher baseline CSF neurofilament light concentrations were more predictive of global and widespread white matter changes and diffuse cortical grey matter changes, which this distinction fits well with what we actually know biologically about NFL, because we know that NFL is a structural axonal protein, reflects global neuroaxonal injury, whereas PENK appears to be more linked to striatal pathology. What was also quite interesting was actually we saw some differences between CSF and plasma NFL in these very early disease stages. And in terms of the analysis, sort of baseline plasma NFL was associated with more of a restricted pattern of brain volume loss, limited to small clusters in the cortical grey matter, which was quite different compared to what we see with CSF NFL. And this finding particularly suggests that maybe plasma NFL could be less sensitive than CSF NFL for detecting early neurodegenerative changes in these far from onset cohorts. But nonetheless, there is still utility and use in NFL in this context.
So as you mentioned, they are biomarkers of a slightly different process, one more specific, one more linked to the striatal neurons, one more general, just reflecting neuroaxonal damage. How do these biomarkers can inform about the pathophysiology and the neurodegenerative process at the earliest stages? Should we stick to proenkephalin? Should we use both to have complementary information? What do you think about the use of both biomarkers?
That's a really good question. And rather than thinking of them as competing biomarkers, I think they reflect complementary disease processes. So PENK is more of a signal of striatal specific neuronal vulnerability, whereas NFL captures broader neuronal axonal injury. And when we actually looked at the disease staging, so I talked about how the cohort was predominantly stage 0 and 1 before clinical signs. When we did receiver operator curves to look at the sensitivity of PENK, we were comparing to basically see whether CSF PENK is similar or could outperform NFL in distinguishing between these two stages. And we know that stage 1 is defined by evidence of striatal involvement, caudate and putamen atrophy loss. So it would be biologically intuitive that a striatally enriched biomarker such as PENK could fit in with this transition. And when we did this, we showed that PENK was actually more superior than NFL in distinguishing stage 1 from stage 0. And when you combined both biomarkers, there was a modest improvement in this discriminatory performance. So I think overall, one of the most key challenges in HD is that by the time motor symptoms emerge, there may have been substantial, largely irreversible striatal neuronal loss. And when there's no clinical signs, for example, in stage 0 and 1, disease monitoring really depends on objective biological measures. And that's where the biological and neuroimaging biomarkers come in. PENK has particular potential in this context because it reflects striatal neuronal integrity and it could predict future striatal atrophy. I think both NFL and PENK in combination offer a more sort of nuanced and biologically grounded picture of these early Huntington's disease changes that are occurring.
That leads to my next question. So I understand that this biological staging system of Huntington's, the progression between a stage 0 when there is no detectable neurodegeneration and a stage 1 when there is and no symptoms, but evidence of neurodegeneration. This is demonstrated usually with MRI. And my understanding is that not wet biomarkers are included in this staging system. So in terms of future clinical trials, how do you think the results of your study and in general biomarkers on NFL and phalin and other potential wet biomarkers, how do you think they will inform the design of clinical trials? How do you see your results will be used in the near future?
That's a really good question from a sort of planning of future preventive clinical trial design. And like you say, the current HDISS criteria don't incorporate wet biomarkers. So I think potentially the potential to expand the HDISS criteria for stage 0 and 1 to where we could potentially include wet biomarkers such as PENK and NFL to further stratify and subdivide stage 0 and 1 could be useful. And from a preventive trial design aspect, having adjusted norms and more populative data of these biomarkers across the norm, we could potentially have staging enrichment, which could be useful for clinical trial recruitment.
Excellent, yeah. And I think recently the news have reached the general media, and I think Huntington's disease in terms of prevention and disease-modifying clinical trials is probably a few steps ahead of some of the other neurodegenerative movement disorders with genetic therapies and other successful studies that have been published so far. Just my question, where do you think that the future of clinical trials in Huntington's disease is heading? And how do you see your results informing this? And what do you think will be the next highlight of Huntington's therapeutics?
Yeah, Huntington's disease, the therapeutic landscape has expanded considerably over the last 5 to 10 years. Lots of different approaches. So beyond Huntington lowering, there's splicing modulators, there's viral vector gene therapy, and more recently, somatic CAG repeat expansion being a target as well for disease-modifying therapies is in the pipeline. I think while current clinical trials are in phase 2 with some in phase 1, I think the aim and hope would be for these programs to expand into phase 3 trials onwards, and hopefully with the disease-modifying trial having approval via sort of regulatory bodies. But I think while all that's going on, thinking ahead of future preventive trial design is really important if we want to try and prevent or slow down disease progression in clinically asymptomatic and pre-symptomatic cohorts, such as individuals that are in HDISS stage 0 and 1. And I think in order to make steps towards that, that's where longitudinal observational studies like the young adult study are quite important, because in order for regulatory bodies really to accept biomarkers as surrogate endpoints, they need to be deemed reasonably likely to predict clinical benefit. And, when you think about these very early stages, traditional clinical endpoints, such as the composite UHDRS, which is currently used as an endpoint in current clinical trials of stage 2 and 3, those endpoints are not relevant for stage 0 and 1 when there are no clinical signs. So that's really where objective biomarkers such as biofluid biomarkers and neuroimaging markers are really important.
Thank you very much. I think it's like a very promising landscape for Huntington's disease research in the next years ahead. Thank you very much, Mena, for your time today. I had Mena Farag from the UCL Queen Square Institute of Neurology in the Huntington's Disease Center. I hope you find the podcast interesting, and I would encourage all the readers to read the full paper published in Movement Disorders Journal. Bye-bye for now.
Thank you.
If you enjoyed the conversation about this paper, you can further your experience with continuing medical credits, or CMEs. You can find the link to the journal CME course for this paper within the episode description on the MDS website. Journal CME is planned and implemented in accordance with the accreditation requirements of the Accreditation Council for Continuing Medical Education, ACCME. The International Parkinson and Movement Disorder Society is accredited by the ACCME to provide continuing medical education for physicians. The International Parkinson and Movement Disorder Society designates this education activity for a maximum of 1 AMA PRA Category 1 credits. The views and opinions expressed by the participants in this podcast do not necessarily reflect those of the International Parkinson and Movement Disorder Society or their affiliated journals, Movement Disorders and Movement Disorders Clinical Practice. Any disclosures of the participants can be found within the episode description located on the MDS website.